And so we move on to the use of E. coli in microbial cloning. Now, there are several reasons as to why E. coli became so widely used and is still a common host for recombinant DNA, and I will be telling you the reasons why. First is their genetic simplicity. Bacteria like E. coli are excellent for genetic research because they have relatively few genes. Approximately, they have 4,400 compared to complex organisms like humans, which have approximately 30,000. Bacteria exist their whole lives as haploids, meaning they only have one set of chromosomes or alleles. And because of that, they become useful in experiments because there is no second allele to mask the effects of a mutation. Next is their fast generation time. Bacteria like E. coli grow extremely fast. A new generation can be produced in about 20 minutes, and this means experiments can yield genetic results in hours instead of days, months, or even years. Faster growth also translates to better production rates when bacterial cultures are used in large scale fermentations. Now we move on to pathogenic bacteria. These are bacteria that form a parasitic relationship with other organisms, and so they are classified as pathogens. They are a major cause of death and disease globally, and they can cause serious infections like tetanus, typhoid fever, diphtheria, syphilis, cholera, tuberculosis, and foodborne illnesses. They can also cause diseases in plants and farm animals, and each type of pathogen has a specific way of interacting with its host. Next, we have the types of pathogens and their effects. First is the common cause of illnesses, like Staphylococcus or Streptococcus, that can cause a range of issues. Next, we have the obligate intracellular parasites, and examples of these are the Rickettsia and the Chlamydiae. Lastly, we have opportunistic pathogens, which can primarily cause disease in people with immunosuppression or existing conditions like cystic fibrosis. We move on to the application of microbes. Microbes have a vast economic importance because they are exploited by humans for many beneficial uses, even though some also play in harmful roles. They can cause disease or they can spoil food. Industrial applications in microorganisms are widely used for the commercial production of numerous products, like in our chemicals, our alcohols and acids, our food, fermented foods like yogurt, cheese, bread, and vitamins and medicines for our health, and finally for biocatalysts, we have enzymes. Microbes are crucial for agriculture. They play a role in nutrient recycling and crop protection, and they are utilized in two main ways. First is they are used as biofertilizers. They are used directly to enrich the soil. They use certain types of bacteria to use as alternatives to chemical fertilizers because they can fix atmospheric nitrogen. They are usually widely produced, particularly in the Indian market, as they help plants get essential nutrients. Their second utilization is aiding nutrient uptake. They help plants absorb nutrients like nitrogen, phosphorus, potassium, and calcium. They are especially helpful for phosphorus nutrition. Lastly, microorganisms are vital for maintaining environmental quality by helping with the biodegradation of waste and pollutants. Their key roles in cleanup include the waste treatment, which in microbes break down urban, municipal, and industrial waste into useful products. For pesticide biodegradation, they efficiently break down harmful chemicals and pesticides. They act as natural purifiers. For heavy metal removal, microbes can be used to remove toxic heavy metals from industrial wastewater, and for oil and grease cleanup. Certain bacteria can metabolize and break down hydrocarbons found in petroleum, and this makes them extremely useful for cleaning up oil spills and grease from water bodies. A strain of Pseudomonas aeruginosa, for example, produces a glycolipid emulsifier that breaks down the oil water tension and they effectively remove the oil.